gonna try to say the name of your PhD university. I'm just gonna say Germany. Because I looked up the acronym and I was like, there's no way. Okay, so your PhD was in plant biochemistry, and then your MS was plant biotech, and then you did a stint in Japan studying microbiology. Okay. I'm currently a research assistant.
Thank you.
We'll give everybody 1 minute to join. And you've got some family joining as well?
I think so.
Okay.
So we have friends and family.
Okay, there was a passcode in the email, so I hope they saw that. Bugs rule, capital B, B-U-G-S, capital R, U-L-E, exclamation.
Excellent.
Yes. Okay. Are you ready?
I can be ready.
Okay, I'll go ahead and introduce you.
Thank you.
All right. Hi, everyone. Thank you for coming to our second seminar of the semester. Today we have our very own Marcin Nowicki talking about the Bradford pear. Hopefully most of you know Marcin. He got his master's in Poland studying plant biotechnology. He also did a stint in Japan where he studied microbiology. He then got his PhD in Germany in plant biotech. And then after that, he had a series of research associate positions both in Poland and in the US. And he has been a research assistant professor here in entomology and plant pathology for a few years now. And he studies the biodiversity of plants, their pathogens. He uses population genetics methods, bioinformatics, and other next-gen sequencing techniques to investigate some of these different relevant problems. And so today he's going to be talking about genetic diversity and population genetics of the infamous Bradford pear. So if you will help me in welcoming Dr. Marcin Nowicki.
Thank you very much for the introduction, and thank you for having me. This is a pleasure to share the progress of one of the more successful research projects that me and my lab have been involved in through the years here at UT. And so I broke it. First slide and I broke it. The computer was logged out, Bia. I will need your help, please and thank you. That's a great start to a seminar.
Sure.
I would like to introduce Dr. Stephanie Mora to all of you. She's sitting us from Brazil. For years from Brabant, Brazil. So make sure you welcome. Yeah. What are you going to work on?
Yeah, thank you for I'm here. Arrived here on August 15th, so it's short ride.
Oh, nice.
And they are now. Is everything nice? That's the morning.
Welcome to EPP. So back to Bradford pear, also known as colored pear, which, as you can see, is almost everywhere, including Knoxville, Tennessee. This picture actually I took on a bike ride when crossing the James White Bridge to South Knox. And it struck me because this population actually is emergent, and this invasive species can be spotted everywhere in the spring. And if you are not color inclined, I think you might be fragrance inclined. Because when in bloom, the tree has a stink. Some people describe it as a rotten fish, rotten meat, owing to the secondary amines that it produces. But also the interesting characteristics is that calorie pear blossoms as essentially first thing after winter. So in a way, it attracts the pollinators by intensive smell, promising them a forage. But calorie pear is just one of invasive species. There is a number of such invasives, and calorie pear might be a good example of what drives such invasions, because the reasons for these species are pretty innocuous. They were very well conditioned, and I will get to that in a second. So what I present here is a list of just 100 most invasive alien species across the world. And yes, insects are important, pathogens are important, but hopefully you will be as surprised as I was when I saw that actually plants are the #1 group of invasives across the world. So this is essentially counterintuitive to what one would expect. And my research and my team's research is helping to solve this issue one species at a time, hopefully, eventually. More interestingly, plants have those complex genomic architectures owing to varied ploidy levels that come from different ploidy tracks, if you will. So by comparison, the mammals, the invertebrates, and other organisms can have smaller and much simpler genomes. So the genomic approaches in plants are much more needed to help cover the gap in the genomic information that we lack to track, to detect, and hopefully to eradicate or control the plant invasives. So the story starts with Shiwani Sapkoda, who joined my lab in 2019. She was the first master student with whom we started tackling the calorie pear problem. And the history of the species is actually inspiring, because I know it feels just like yesterday when early 20th centuries, USDA were sending people to Asia to help combat the Erwinia amylovora, or the fire blight in edible pears, in apples, in many Rosaceae species. So they were essentially looking for rootstocks that would lend the science grafted onto this rootstock the resistance to these bacteria. And one such promising plant was the calorie pear, who comes from Eastern Asia, from China, from Taiwan, Japan, Korea, such Asian region. So this is the origins. We were able to track a number of such original native samples from that region, from both living collections and from herbaria, preserved collections. And that collection actually included one specimen from which the Bradford pear, the name by which the calorie pear is actually known across the US, was developed from. We then expanded this collection for a number of samples from US developed commercial cultivars. And the story is that calorie pear, on top of the resistance granted to whatever species were grafted onto that, had also beautiful ornamental properties. So, of course, people wanted to make extra income on the tree and sell it as ornamentals, the way we know it now. So, in circulation, we have more than 25 cultivars that have been released commercially throughout the years, starting from the 1960s, 1970s, with the Bradford pear. And so we also included for our molecular investigation in this leg of the project, samples of Pyrus and Mylus just as outgroups. So when Frank Meyer was in Asia, early 20th century, he observed that calorie pear actually is a survivor. You can see here that the Pyrus caleriana grows essentially on a sand soil, survives drought, survives flooding, survives a number of pathogens attacks, and yet still blossoms, and yet still allows to be spread across the region, across the terrain. So as such, prolific plant was brought over to US in masses. So there are reports of Frank Meyer sending tons of seeds to US to help the US gardeners for rootstock, for their nurseries, for the apples and their pears. And so shortly after releasing the cultivars, the problems emerged because the trees escaped cultivation. With such masses of inoculum in plants, they were sprouting essentially everywhere. I showed you the James White in 2021, and you can actually see it from satellite images when the plants are in bloom. Such visible, such noticeable difference color repair makes in the landscape. But when we look just a couple years back to 2009, the same area, the same James White bombs were essentially having no color repair. So this is a very fast growing, very fast colonizing plant that essentially can be spotted and can take over the land. So by removing the nutrients, by taking space, it can displace other species, it can outpace the native species, and there comes the problem. I also showed you the huge thorns or spurns that these naturalized specimens are making, not the grafted ones, not the cultivars. Those that actually go in the wild and grow from seeds that are dispersed by birds, they will make the thorns. And such thorns can pop the tires if you have color pairs that are invading maintained forests or natural forests, for which we have documentation in Georgia, in Tennessee, in other states. And a tractor comes in to take lumber, and the thorns pop such tractor tire, that costs the maintenance $1,000 ride for one pop. And this is not uncommon occurrence. This is just another hindrance how the color repair affects us. So the aerial spotting was also a topic of one of my collaborative grants, where we had drones flying over using the infrared cameras trying to distinguish what signatures are specific to Pyrus coloriana. And this research is ongoing, so I will have to leave it at that. But we did get some specific signatures for color repair. Back to Shiwani, because this is all tangents, and I want to focus on the molecular history and molecular disentanglement of color repair. Using the help of EPP scholars, we developed a score of SSRs or microsats, which are molecular markers by their very own inherent feature variable. So they are good tools for us to assess diversity. Shiwani first tested how those markers, 18 of them, cross-amplified to different related species of Pyrus and of Malus. So this is our pears and apples that we had in our collection. And there is substantial cross-amplification, which tells us that this toolbox can be used for similar assessments in those related species to where they cross-amplify. When we take the genotyping data from those 18 SSRs, we end up with a huge spreadsheet of genotyping signals, each of them being one genotype for one particular individual. So this is a multivariate data, multidimensional data. And for human brain to get it, we need to squish it to just dimensions for representation using only the two most representative dimensions. And as you can see here in this DAPC analysis, the first axis and second axis contribute to about 7.4 and 5.9% of the total molecular variability available, discovered, retained in this data set. And then what we see is that the groups, just grouped by origins, are placed in different areas. And so the way to read this graph is that essentially the area for each group would correspond to the diversity retained within this group using our markers. What we see in the upper right corner, though, is a phylogenetic tree built from our genotyping data set. And we here see, in accordance with the historical records, that the group of cultivars that are in sale in the US are the most closely related to the group of native materials from China. And the populations from Korea and Japan are slightly more divergent. Interestingly, when we zoom in on those cultivars, we see something very troubling. For instance, I marked two different individuals of the cultivar Helmsford, and you can see that they should be identical because cultivars are bred essentially by cloning. So you have vegetative clones of the same genotype. So this is what you would expect. And yet, Helmsford is just one example of such possible mishandling of cultivars. So therefore, we asked for trouble from the get-go by not selling what was advertised. Because if a Lowe's come to a nursery asking for 5,000 Bradfords and they have only 3,000 Bradfords, chances are they might be selling something else just to make extra income because this is wholesale. And this information will be coming up again in another slides. So we have a toolbox that we developed and that we validated using these Asian materials native range and a score of cultivars. Qiwan's next project was to see how those markers work actually in the naturalized populations in the northern group in the area of Knoxville and in the southern group on the other side of the Appalachian Mountains, which would be essentially a range, geographical barrier for spread. We hypothesized that those would be related, of course, but that there would be divergence between these two major populations because of this geographical barrier. And so indeed, we discovered a modest relationship between geographical distance and molecular distance. We test this using so-called mantle test. And the mantle test is a regression method that compares two distance matrices. We can use geographical genetic distance. We can standardize it by year of sampling. We can use ecological distance matrix, but it is a mathematical tool that helps us explain what we see in our genotyping data. For our SSRs, we see that only 22%, or if we square it to get the variance explained, about 5% of genotypic differences in the data set of those naturalizing populations can be explained by the geographic distance alone. And this is a pretty often occurrence in such studies. 5% is a healthy, statistically significant result. So when we do the same thing we did for the native populations, when we try to plot on the DAPC our invasive populations, we indeed observe that we have two major clades, one for this southern group and one for the northern group. But another information we draw from our data is that actually the northern group is much more dispersed, much more divergent. So perhaps there are better conditions for those northern populations to thrive, to develop, to spread, to mutate, and colonize new regions. So this is important information from the control standpoint. If we want to focus our efforts, those emergent populations, those new colonizing genotypes with higher diversity might be of more interest to figure out the molecular mechanisms behind the spread of these species. Population genetics is a powerful toolbox. It also lends itself to evolutionary history of the species and trying to figure out what the lineages, how the lineages are correlated. So when we start from the bottom of this graph, this is the current time point. And then we go back in time into the coalescence to see how different scenarios are comparing, what are different times of splits, and how the history is shaped by different trees, the evolutionary trees. So when we combined the genotypic data from Shiwani's first study with the second study, we came up with the highest support for this tree that I'm showing you. And in this tree, we clearly, again, in accordance with historical reports, see that the originating group was the individuals from Asia, from the native range, and a group of cultivars that we genotyped. But then the first diverging population was actually the one on the southern side of the Appalachian Mountain. And from this population, the northern group diverged. More interestingly, we can actually calculate the times in generations of each event. So we know by the species biology that from seed to flower, it's 3 years. If we have roughly 120 years since introduction, we would expect 40 generations. And our results show us that actually the calculated time is 100 times more than that, which means that the introduced materials were recombining and shuffling their genomes many times over until they finally escaped cultivations. And the final result that we can get from this particular tool is the mutation rate per SSR locus and per generation. Working in EPP with population genetics for 7 years now, I have never seen such a high mutation rate. So this mutation rate of 2% per locus per generation is insanely high. We normally, for SSRs, expect in the range of 2 to the power of -5 to the power of -4. So this is 100 times over again, which just underscores the high evolutionary potential of this invader. So this leads to the next project that we started pursuing. If you know of anybody with monetary funds to help us figure it out, I would be grateful. We sampled across the Eastern US. We've got about 1,300 samples of DNA from 125 collection sites that were graciously provided to us by a horde of scholars and colleagues and collaborators. We know that the invasion point was in Maryland because that's where the USDA station is, who actually introduced those cultivars in the beginning. But what happens at the genome level across the colonized range, we do not know yet. Next up, we followed something that is intrinsic to the Rosaceae. Rosaceae have a self-incompatibility system. And this system is essentially preventing a pollen that has a signature identical with the ovule from pollination. This is based on the enzyme called sRNase, and this is essentially protein-to-protein recognition system, again, that prevents pollination. So the undergrad, Sam Conrad, who worked with me since May of last year, was adopting the sRNA genotyping system from the edible pair, and it worked very nicely for us. She established this system by using a generic sRNA-specific markers, and you can see that these two primers that I'm showing you on this slide encompass a lot of this gene. But interestingly, we see disturbances in this sRNA pattern. So, for instance, you can see that you have all sorts of things happening in Bradford. And by the way, these are the acronyms for different cultivars of color repair that we have tested. And you can see all sorts of misfingerprinting. Again, we would have expected the same signals throughout the entirety of a given cultivar, which is very much not the case. To underscore this point further, Sam also used a number of allele specific. So these sRNAs come in different flavors, in different variants. We can also genotype using allele specific markers, and she did the same thing, which resulted in the same observation. that probably there was something happening at the sales point or at the nursery point, that some cultivars were labeled as different cultivars, and that's why we see not true to type, essentially throughout the range of the cultivars that we have tested. So in total, she has used 17 markers. That group includes 9 allele-specific markers, and we have Sanger sequenced a score of those as well. And then we moved on to analyzing the Asian group and the escapes that Shiwani was analyzing as well. So here we see the APC again, and we see that there is substantial diversity throughout all of those groups. If we zoom in on the US cultivars again, we again see the same pattern, that something worrying happened with the cultivars, that we cannot say which of the trees is Bradford, which of the trees is a true aristocrat. The next step is the computer list. Yes, there we go. Dabbling in the genomics started with constructing the absent at that time chloroplast genome for this species. And so what was reassuring was that Choleriana plastidial genome places with all the other Pyrus species of the Asian origin, because the genus has 3 geographical origins. One is in Asia, the yellows are in the Mediterranean region, and the blues are in Northern Africa and Europe. So we were happy to observe that what we've done seemed correct, but then we can take it further. By pulling the genomic data from the databases from NCBI, we can discover how the chloroplast genome is diversified, how many variants are across this genome. And there is a number of those variants. And just to prove it, we chose 21 of such variants, because we had so many enzymes in the lab, so we didn't want to make it too big of a deal. And the same happened with mitochondrial genome, where, because it's substantially larger, also has many more variants to possibly analyze. So Will Baseman, the undergrad who was working on it over the summer, analyzed those 14 regions, but in 21 different reactions, because several of the regions had single cutters using 2 or 3 different enzymes. So we can essentially stack the information and save time by doing the PCR one time, but cutting in 3 or 4 different ways. And he did observe similar things to what Sam and Shiwani have seen, also in the plastidial genomes. So we see, for instance, here a lot is happening in Bradfords, including different variants, different alleles of chloroplasts. And the same things happen, for instance, in the Holmesford at the bottom of the slide. Again, we observe that not true to typeness is present throughout our dataset. Finally, Alina joined the lab last year, and she is working on a project that was graciously funded by the USDA Forest Service. Her work is to help enforcing the ban of sales of colored pear that is being implemented in South Carolina, in Ohio, and in an increasing number of states. We want to use LAMP and LAMP loop amplified, I'm drawing a blank, or what is the full name, which is a PCR-based method, uses 6 different primers to essentially cover a detection locus in different ways, so that when the PCRs are built on that, it kind of helps build up itself in a very fast way. As long as we have a specific locus, we can do wonders with it, and Alina actually did. First, we started off with alignment of the plastidial genomes. Choleriana is hidden because it's the baseline. So when we compare Choleriana with Betulifolia, Perifolia, and Communis, the 3 species that are most often grafted onto the seeds of Choleriana, we see a number of promising loci. Whenever you see a dip, This is essentially a marker of a nucleotide difference from coloriana, the baseline, to our species of interest. And we saw a number of such positive prospective spots for Alina to explore. And so we recovered 19 potential targets throughout the plastidial genome of coloriana. And then we only were able to develop 9 LAMP primers because LAMP comes with a number of requirements need to be met. And the idea was that detection of coloriana might not need specialized skills because unless you're colorblind, you can distinguish a color change in the reaction between red, which is negative, to yellowish or green, which is a positive reaction, equals detection of coloriana. Another option is that turbidity is also indicative of positive reaction. So even if you are this unfortunate person with color blindness, you can still see what is happening in your reaction. So to start things off, Alina was very busy sequencing her locations to make sure that we see differences. And we did see differences between coloriana and other species of target. And then Alina optimized her LAMP detection towards a field tool, which can be applied by such portable PCR machine that you can use. You can power up with batteries, you can power up with your car. So a field agent with no specific master's or PhD in molecular biology can take this tool to a nursery to see whether they observe the sales ban of coloriana. So what Alina came up with was actually stunning. She was able, by applying serial dilutions in one of the mitochondrial loci, come down to just 0.5 picograms of DNA. And this detection equals 4 copies of the target sequence, which in practicality means 1 cell that is needed for successful detection. And this detection can take, as you can see on the vertical scale, as low as 25 or 30 minutes. So within minutes, you can have an answer whether or not a salesman is observing what the law wants to. The other important take from Alina's research is that we actually do not need to even isolate the DNA in the lab. We can just take a leaf sample, a root sample, a flower, or a bark, grind it in buffer, and apply this buffer to PCR in the field, in the nursery, in a shop. You can see that without dilution, there are problems which may signify that there will be inhibitors that we are extracting alongside. But if you dilute it a couple times, the problem is gone and you get beautiful detection down the road. So finally, this brings me to another observation, another future possibility for research, which is that colory pear is an ecological desert. There are virtually no pathogens, no insects that would eat it. It serves barely as emergency food for birds that have nothing else to find in the winter. And that means that must be something in the colory pear that prohibits growth of pathogens or feeding of insects or deters their feeding on. So if we can apply a series of isolations from whatever organ tissue we choose, and then try growing different pathogens on such enhanced media, we can actually detect what biochemical defenses colory pear can lend us to help leverage its presence in the land. Again, this is just the future, and I am very excited about this possible future opportunity. Just to bring you up with the wrap-up, this research happened because of essentially small army of people, and we were able to push a number of deliverables throughout since 2019, and many more are to come. And at this stage, I would very much like to thank the helpful EPP and UT for having me and for letting me explore all this diversity and applications thereof. and of course, the group of people who worked on it, including undergrad and grad students, and Janita, who was actually Dr. Hajabich, who was the first person of contact for this project. This is how science works in 21st century. You can get contacted by David Coyle from Clemson over Twitter, asking for help, and that's how the magic happened in this case. Or you can get invited by Dr. Bob Trujillo, start working as a postdoc, and then come up with this beautiful strain of projects that I am very happy that you are paying attention to. Thank you for your time, and I'm very happy to take your questions.
So you mentioned that there's not that many insects that feed going there. Is this true also in its native range?
The question from Dr. Fuentes was because of this allegation that in US, Caloripair is not being fed on by insects, whether the same happens in Asia. That's a great question. In Asia, actually, Caloripair is on the brink of extinction. It is on the list of endangered species both in Taiwan and Japan. And there is decreasing number of observations of this tree in China. So in essence, we don't know.
Something is affecting them.
Right. So something must be happening to the tree in the native range that is not here, that allows it to thrive.
We'd be happy to send some back.
Do you think that the mishandling of the cultivars and misrepresentation, is that just from ignorance or is it outright deception? Like, do you have any? thoughts
Yeah,
on
I am disinclined to discuss politics in a public setting. But if you think about it, the motives are actually very troubling, because if this happens for a beautiful tree that everybody wants to have in the gardens or line the parking lots, this may very well have happened for many other species that we have in circulation, right?
Right.
So I guess the major take-home message from this talk would be to be careful what you wish for. If you want to have an exotic plant in your backyard, you may want to think twice.
Can you talk about, you were talking about doing a PCR out in the field? What system were you guys using for that?
Dr. Charity Owens asked about PCR system in the field. So we will be applying the lamp, which proved to be working really fast.
Okay, and that's something you can do in the field.
That's the idea, right? I showed you the picture of this mini PCR machine that actually is very affordable. Yeah, they are not paying me enough for this. It is very affordable, and you can equip it, and you can take it to the field. And as I explained, you can just grind a tissue and do it in the field within half an hour by external power source, by connecting it to your car battery. So this is a portable machine, and because of this portability, which is such a huge deal for field agents, we think that LAMP is uniquely suited because of this the amplification that is isothermal, essentially. All you need is to maintain 60 degrees in the chambers, in the tubes for half an hour. So this is not a complicated system, whereas PCR needs up and down so many times.
Right.
I'm curious about the way that the chemicals is extracted in that PCR reaction field. You're using like leaves and roots and different parts. It seems like they all work well, but I'm a little surprised because I would expect that some things are harder to extract in your material than others. You use like a solution that's very effective.
Yes, Dr. Fuentes asked about the tissues that we use for field detection. So I guess the idea behind all that is to see whether we can do it first. If we can do it, then the second question becomes, what is the most sensible thing to do? In the context of color repair and in the context of controlling the sales whenever the bans are invoked, we would actually test the rootstock to make sure that rootstock is not of the color repair origin. And taking a single roots and grinding it is actually doable, especially if these are younger roots. And rootstock all come from one single seed. And on that, whatever is grafted is then being sold as a cultivar or cultivated one pair or another or different species. So the younger roots lend themselves very nicely to such a process. And Alina has done it in a 2 ml tube with a cutting of such root that she weighted carefully just for us to see what range is applicable, and a little bit of the elution buffer from the DNA kits. So yes, it worked, and it seems like a very plausible way to go forward.
Do you have any updates on the bans throughout the US? Are there more states that are going to adopt that?
Dr. Owens asked about the bans throughout the US. To my knowledge, there are only 2 states that have banned it.
Okay.
And that was also why the USDA Forest Service liked our grant, that we would help invoke these regulations further. The presence of color repair as naturalized or invasive species has been confirmed in, I believe, 35 eastern US states, but only 29 as of spring this year put color repair on their respective lists of invasive species. And only 2 of these have now the sales bans.
So you can still find calorie repair like at a Lowe's or a Home Depot and other.
Absolutely, you can. To a lesser and lesser extent, I had troubles buying color repair cultivars from Lowe's and from Home Depot. I had to call a nursery in Georgia, and I will not give you the name because I do not want it to spread further. But yes, it took me a long time to actually track places that do sell it nowadays, even in Tennessee.
A lot of these places that have, at the very least, this on their invasive species list, do they have incentive programs for people to get rid of their calorie pairs in their yards and on their property?
Okay. Dr. Owens asked about incentives for getting rid of color repair. Such programs have been implemented by David Coles in Clemson and other people. And I think they are kind of instrumental and inspirational, really. Throughout the years, what they have done, they offered people who showed them picture of the color repair that was put down from the front yard, and they gave them a sapling of a native tree to replace it. So the first year was during COVID, so, but the further years, this program actually enjoyed a lot of attention, and there were hundreds, if not thousands of people, to the point that during the first day of this replacement, they ran out of the trees to provide to people. So I guess it was a good stretch goal. It was a good incentive.
Do you think it helps as far as reducing?
Well, seeing the map that I showed you throughout the eastern US, I guess the genie definitely is out of the bottle. But perhaps anything we can do helps. And I want to think that, every penny counts in a way, that every tree that is removed will no longer provide annual seeds to spread away again. So as much as we can postpone the invasion throughout the US would be nice.
Any other questions? Do we have any questions on the chat? All right. Thank you, Dr. Novitsky. That was a great presentation.
Thank you very much, everybody.